SANDHAN, All Gujarat Integrated Classroom.
સેટેલાઇટના માધ્યમથી જોડતી કડી એટલે સંધાન.
Hello, good afternoon viewers. Today we are here to talk on a topic which is related to the two surface appendages found in the bacterial cell. We are going to consider the structure and function of the two important appendages, namely Flagella and Pili. Myself is Sneha Pandya from MG Science Institute, and with me is Piyush Pandya from Gujarat Arts and Science College. And today we will try to give you the information. Primary information will be covered by me and the detailed information will be covered by Piyush. So let us start. The first slide, as you can see, this shows the typical prokaryotic bacterial cell structure. As all of us know very well that prokaryotic cellular organization is the simplest cellular organization found in the biological systems, and various structures have been displayed in this prokaryotic bacterial cell. We are going to highlight the two important structures which we know them as the surface appendages. As you can see over here, the one which is shown singly and which is a long wavy structure is the flagellum, shown brown in color, and you can see the presence of numerous shorter structures present all over the cell surface, and they are pili. The same has been shown here in the next photograph, where you can see the presence of number of flagella, which are long hair-like wavy structures present at the pole of the cell, whereas the smaller structures, fimbriae, as have been mentioned over here, they are present all over the surface. At this stage, I would like to say that the two terms pili and fimbriae, they are synonymously used with certain exceptions, where some special type of pili which are involved in some other important function, for them this term is not applicable. Generally this is applicable for the somatic pili which are there and which are responsible for giving the property of adherence to the bacterial cell. In the next slide you can see, now let us have the basic information about the flagella first. As it has been mentioned, the singular is flagellum, whereas the plural is flagella. These are the long wavy ape-like structures, and chemically they are made up of a protein which is flagellin, which is known as flagellin. We know that flagella are the locomotory organs of the bacteria, and they are responsible for the movement of bacteria. Flagella can rotate either in clockwise or anti-clockwise direction, and as a result of that the movement of the bacteria takes place. As it is known that the bacterial movement mostly is observed in aquatic environment, that is in presence of water, where relatively low viscosity is present, and bacteria, they show movement in response to the stimuli. The movement which is there as a response to some stimulus is described as tactic movement, and depending upon the stimulus, for example, if it is light as a stimulus, then the movement is described as phototaxis. If it is some chemical, it is chemotaxis, and if it is magnet, it is magnetotaxis, and so on. So in short, bacteria, they move or they show movement in response to the stimuli. In the next one, we have given the general information about the pili. Plural is pili, singular is pilus, and it is mostly found in gram-negative bacteria, and these are comparatively shorter and straighter structures than bacteria. They are tubular and they are also made up of a protein which is known as pili. Now, in the next slide, as has already been said, these are comparatively shorter, straighter, and they are sticky and bristle projections present all over the cell surface, and these are basically there to give the property of adherence to the bacterial cell. With the help of the pili, bacteria, they can adhere to another bacterial cells. They can also have adherence with their host cells. This is very important in case of pathogenic organisms, and they can also give the property of adherence to the bacteria to their substrates in the environment. And exceptionally, there are some special types of the pili which are also involved in the transfer of the genetic material from one cell to another. In the next slide, as I have already told you, the pili which are basically there for attachment, these are also known as attachment pili or somatic pili, and they are there to give the property of adherence, whereas the other type of the pili, special type of the pili, these are known as sex pili, and they help the transfer of the DNA from one bacterial cell to another during conjugation. How the event takes place, that we are going to cover later on in detail. Now, let us see about the arrangement of the flagella. We know that the bacteria which are flagellated, that is the bacteria which are having presence of flagella, the development and the arrangement of the flagella on the different types of the bacteria is also different, and it is the particular or it is specific for the specific species. And the development and distribution of flagella on the bacterial cell surface has been considered as one of the important criterion for the classification of the organisms. Now, let us see how the arrangement of the flagella is there on the bacterial cell. These are described as the monotrichous if the single flagellum is found. They are lophotrichous if the tuft, that is the bunch of the flagella is found at end of the cell. It may be amphitrichous when the flagella are found to be present at both the ends, and it would be peritrichous when they are found all around the bacterial cell surface. So, this is an important characteristic, important parameter or important point to be noted. Here in this figure, you can see the long wavy hair-like structures, flagella, how they are there present at the particular position. In the first, it is mono, single flagellum is there at one end. In the second one, there is a tuft of flagellum, bunch of flagella present at one pole. In the third one, a single flagellum is present on both the poles, both the ends of the cell. And in the last figure, it is the flagella, number of flagella are present all over the cell surface. Here you should note that in the first three figures, mono, lopho and amphitrichous, the flagella are present at either of the poles of the cell. They develop and they are present there at the end of the cell, at one of the ends of the cell. In case of the fourth figure, the flagellation is there found all over the surface, that is the lateral flagellation is there along with the polar flagellation, and as a result of that, finally it is the peritrichous flagellation where large number of flagella are found to be present all over the cell surface. The same here, the color figures are there showing the different patterns of the arrangement for the flagella. In the next one, this is the SEMs, scanning electron micrograph, showing the various patterns of the flagellation. Here I would like to say that this type of the photography. It is the false colored type of the photography. You should not take this as the colored microbial cells. These are not colored microbial cells. This is the simple SEM showing the various patterns of the flagellation. And you should also be knowing that while observing the movement under the microscope, ordinary microscope that we use in the laboratory, we can see the movement of the bacteria, but we can't see the flagella even though they are there. And that is due to the extremely small sized, that is the extremely small diameter of the flagella, which falls below the limit of the resolution of the light microscope. And so flagella we cannot see, even though we are able to see the movement. And to see the flagella, we know that the special type of the staining techniques are required, and in then we can see the flagella after staining them with the suitable technique. In the next one, we are going to consider the different the detailed structure of the flagella, where here you can see the description is there about the different regions of the flagella, and all the details will be given to you by Piyush. So please.
Namaskar. Good afternoon, everybody. The background is already has been laid, and now let us talk in detail about the structure of the bacterial flagella. As already known to most of the students, the flagellar body itself can be divided into three different portions, starting with the basal body and then the hook and the filament. What we usually identify as the flagellum in diagrams and in figures and in photographs is actually only the third and external part of the flagellar structure. The three-phased structure contains the basal body, as I said, and then the hook and then the filament. What we see external to the cell is only the filament part, and that too, as Sneha has already mentioned, can be observed only if it is properly stained or has been given some other treatment so as to increase its width. Otherwise, in its natural diameter, the flagella or flagellum rather is not possible to be visualized using an ordinary light microscope. As the student fraternity must have always noticed, they would have seen the motile bacteria moving here and there quite often under their high power objective using the hanging drop preparation. But have you ever wondered why the flagella cannot be observed? The reason is known to you, students. The diameter of the flagella is so restrictive to the limits of light microscope that it cannot be observed. Anyway, that is the external part. The internal part, as I said, is known as the basal body, and the basal body and the filament, the external part, are connected with each other through what is known as a hook that acts as a mediator between the two. As this slide shows, the first part, the helical filament, is, as I said, what is observed externally. It is a long region and constitutes almost 90% of the total length of the flagellar structure. The rest 10% is constituted by the hook and then the inner basal body. The filament part of the flagella is made up of a protein which is called flagellin protein, and as we will see later in a video which we have, the flagella has a tendency of growing at the tip. That is, when the flagellar growth takes place, the flagellin molecules, individual flagellin units, are always added into the growing filament structure such that the latest body goes and places itself at the tip of the flagellum. So when you look at the tip, that is the part which is synthesized at the latest. We'll look at that video later on, not at the moment. Now these flagellin bodies are constituting into a single filament structure known as flagellum or the filament. Then, as we have already talked, the next is the hook, which provides the link between the filament and the basal body, and the part of the flagellar structure which is probably the most important for the movement of the flagellum and thus that of the cell is the basal body. The basal body, as you can read in the slide, is the terminal portion of the flagellum which falls inside the cell wall and cytoplasmic membrane, and it fixes the flagellum to the cell wall and the cell membrane. There is a central rod surrounding which there are certain discs present. Now the number of the discs is always dependent upon the kind of bacterial cell. If it is gram positive cell, there are only 2 discs, and if it is gram negative cell, there are 4 discs. We will be elaborating upon this looking at this diagram. As this diagram shows you, the structure on your left is that of a flagellar body of gram negative organism. You can see there are 4 different disc-like structures observed surrounding a central rod, and then there is a hook in between, and external to everything else is the filament. Now look on your right hand side, the structure of flagella in case of a gram positive cell. You can see what is conspicuous by absence are those 2 external discs which are seen in the basal body structure of gram negative organisms. The reason why gram negative organisms have 4 discs and gram positive organisms have only 2 is very interesting. The lower 2 discs, which are observed both in gram negative as well as gram positive organisms, are called the S and the M disc or S and the M rings.